Hello everyone, this is Sai speaking. Today in this video we are going to learn about restriction endonucleases. Now, what is restriction endonuclease? A restriction enzyme is a protein isolated from bacteria that cleaves DNA sequences at sequence specific sites, producing DNA fragments with a known sequence at each end. The use of restriction enzymes is critical to certain laboratory methods, including recombinant DNA technology and genetic engineering. Now, coming to the history of restriction enzyme, the term restriction enzyme originated from the studies of enterobacteriophage, that is lambda phage, in laboratories of Werner Arber and Matthew Wilson. The ability of certain E. coli strains to inhibit the activity of lambda phage by the enzymatic cleavage of the phage DNA was studied, and the enzyme responsible for its growth restriction was termed as restriction enzyme. Werner Arber and Daniel Nathans and Hamilton O. Smith were awarded the Nobel Prize for Physiology or Medicine in 1978 for their discovery and characterization of restriction enzymes, which led to the development of recombinant DNA technology. Now, coming to the brief part of restriction enzymes. Restriction enzymes are also called molecular scissors as they cleave DNA at or near specific recognition sequences known as restriction sites. These enzymes make one incision of each of the two strands of DNA and are also called as restriction endonucleases. Viruses infect the host cells by injecting their DNA into the cells. This viral DNA hijacks the host cell's machinery for reproduction of viral progeny, resulting in host cell's death. To overcome the viral infection, many bacteria and archaea have evolved several mechanisms. A major protective mechanism involved in the use of restriction enzymes to degrade the invading viral DNA by cleaving it at specific restriction sites. At the same time, the host cell protects its own DNA from being cleaved by employing other enzymes called as methylases, which methylate adenine or cytosine bases with host recognition sequences. For each of the restriction enzyme, the host cell produces a corresponding methylase that methylates it protects the host DNA from degradation. These enzymes make up the restriction modification systems. The restriction enzymes catalyze the hydrolysis of the bond between three oxygen atom and the phosphorus atom in the phosphodiester backbone of DNA. The enzymes require magnesium plus 2 or other divalent ions for their activity. Now, coming to the nomenclature, Smith and Nathans suggested the naming guidelines for restriction endonucleases in 1973. According to these guidelines, the names of the enzymes begin with italicized three-character acronym. The first letter indicates the first letter of the bacterial genus from which the enzyme has been isolated, and the next two letters are derived from the bacterial species. These may be followed by extra letters or numbers indicate serotype or strain. This is followed by a space and a Roman number to indicate chronology and identification. For example, HIND3 was the third of four enzymes isolated from Haemophilus influenza stereotype D. Now, in this image, we can see that of E. coli, E describes about genus that is Escherichia, and CO describes the species name is coli, and R is the strain name RY13, and 1 is the order of identification. It is the first identified. Now, coming to types of restriction enzymes. Restriction enzymes are traditionally classified into four types on the basis of submit composition, cleavage position, sequence specificity, and cofactor requirements. However, amino acid sequences have undercovered extraordinary variety of among restriction enzymes and revealed that at the molecular level there are many more than four different types. Type 1 restriction enzymes. Type 1 enzymes are complex, multi-subunit, combination restriction and modification enzymes that cut DNA at random far from their recognition sequences. Originally thought to be rare, we now know from the analysis of sequenced genomes that they are common. Type 1 enzymes are considerable biochemical interests, but they have little practical value since they do not produce discrete restriction fragments or distinct gel binding patterns. Now, coming to type 2 enzymes. Type 2 enzymes cut DNA at defined positions close to or within the recognition sequences. They produce discrete restriction fragments and distinct gel banding patterns, and they are predominant class used in the laboratories for routine DNA analysis and gene cloning. Rather than forming a single family of related proteins, type 2 enzymes are a collection of unrelated proteins of many different sources. Type 2 enzymes frequently differ so completely in amino acid sequence from one another, and indeed from every other known protein, that they exemplify the class of rapidly evolving proteins that are often indicative of involvement in host-parasite interaction. Most common type 2 enzymes. The most common type 2 enzymes are those like HAL, HIND3 and NOTL that cleave DNA within their recognition sequences. Enzymes of this kind are principal ones available commercially. Most most recognized DNA sequences that are symmetric because they bind to DNA as homodimers, but a few recognize asymmetric DNA sequences because they bind as heterodimers. Some enzymes recognize continuous sequences in which the two half of the recognition sequence are adjacent, while other recognize discontinuous sequences which in the half sides are separated. Now, type 2S enzymes, the next most common type 2 enzymes usually referred to as type 2S are those like FOK cleave outside the recognition sequence at one end. These enzymes are intermediate in size, 400 to 650 amino acids in length, and they recognize sequences that are continuous and asymmetric. They comprise two distinct domains, one for DNA binding, other for DNA cleavage. They are thought to bind to DNA as monomers for the part, but to cleave DNA cooperatively through dimerization of cleavage domains of adjacent enzyme molecules. For this reason, some type 2 enzymes are much more active on DNA molecules that contain multiple recognition sites. Now, type 2G enzymes. Type 2G restriction enzymes are third major kind of type 2 enzyme are large component restriction modified enzymes 850 to 1250 amino acids in length, which the two enzymatic activities reside in the same protein. These enzymes cleave outside of the recognition sequence and can be classified as those that recognize continuous sequence and cleave on just one side. Now, type 3 enzymes. Type 3 enzymes are also large combination restriction and modification enzymes. They cleave outside their recognition sequences and require such sequences in opposite orientations with same DNA molecule to accomplish cleavage. They rarely give complete digestion. Type 4 enzymes. Type 4 enzymes recognize modified, typically methylated DNA are exemplified by the MCR-BC or MRR systems of E. coli. Now, the factors affecting the activity of restriction enzymes. Depending on the substrate DNA and the reaction conditions, restriction enzymes show a wide variation of cleavage and possible star activity. In order to obtain the desired cleavage, it becomes more important to control the following factors. Star activity. Under suboptimal reaction conditions, some restriction enzymes cleave base sequences at sites different from the defined recognition sequence. In other words, they cleave at non-specific sites. This phenomena is called star activity. The methylated DNA. Several DNA molecules are methylated at recognition site, making them resistant to cleavage by certain restriction enzymes. For example, most E. coli strains are DAM or DCM methyltransferases that methylate specific recognition sites from G6M80C and C5MCA or TGG, respectively. Temperature. Most endonucleases optimal digest the target DNA at 37 degrees Celsius. However, there are some exceptions with lower and higher optimal temperatures. For example, TAC1 optimal dies at 65 degrees Celsius and APA1 dies at 25 degrees Celsius. Restriction enzyme digestion. Restriction enzymes recognize a specific sequence of nucleotides and produce double-stranded cut in the DNA. The recognition sequences can also be classified by the number of bases in its recognition site, usually between 4 to 8 bases, and the number of bases on the sequence will determine how often the site will appear by chance in given genome. Example, 4 base pair sequence would theoretically occur once every 4 into 4 or 256 base pairs. 6 base pairs is 4 into 6, 4 power 6 or 4096 base pairs, and 8 bases would be 4 power 8 or 536 base pairs. Many of them palindromic, meaning the base sequence reads the same backwards and forwards. Restriction digestion of double-stranded DNA produces two kinds, sticky ends and blunt ends. Blunt ends possess a 5' phosphate group that promotes ligation. They are universally compatible with other blunt-ended DNA. For example, E. coli. If you see the diagram here, the it cleaves at GAT and ATC. Different. Now coming to the stick ends, they are small stretches of single-stranded DNA capable of self-ligation or ligation with completely complementary region from other DNA molecule. The sticky ends possess 3 or 5' overhangs of 1 to 4 nucleotides. In the diagram of VLN1 we can see that. Now coming to the isoschizomers and the neoschizomers. Isoschizomers are restriction enzyme with the same recognition sequence and cleavage sites. And neoschimers are restriction enzymes with same recognition sequence but cleave DNA at different site with that sequence. Now coming to the application, there are several applications with restriction enzymes. Now coming to molecular cooling, the popular application of restriction enzyme has been generation of recombinant DNA molecules. The process involved in the cutting of donor DNA and the vector DNA by restriction enzyme to yield compatible ends. Now, DNA mapping, also known as restriction mapping, involves in the use of restriction endonucleases to obtain structural information of DNA fragment or genome. Mapping involves determination of the order of the restriction enzyme sites in the genome. Restriction landmark genomic scanning is a genome analysis method that utilizes a combination of restriction enzymes to visualize differences in methylation levels across the genome of a given organism. It is useful technique to identify deviations from a normal in any DNA. Now coming to the gene sequencing, a large DNA molecule can be sequenced by digesting within restriction enzyme and processing the resulting fragments through a DNA sequencer. Thank you everyone for listening to me. If you are having any doubts, feel free to ask in the comment section. I will try to clarify. Thank you again.